revisit the subject of Chapter 1, blood flow in and out of ampulla attached to a colony. This colony has 3 developing zooid buds on its side, indicated by the 3 black ovals. The smaller rectangle indicates the peristaltic heart, which you will see beat. This video is speeded up 20 times. Blood flows in and out of ampulla and through the vessel net under the body. Flow is synchronized on the upper right side and the lower left side, but they are 180 degrees out of phase. Body size doesn't change much. Blood exits one side while it enters the other. This pattern varies among colonies. Now go back to the end of Chapter 2, when we looked at contractions of a detached single ampulla. It looked like the contractions progressed down the axis of the ampulla. I plotted area versus time for the 4 quarters of the ampulla, as illustrated here. These 4 sine-like curves all have the same period, but are shifted in phase. Quarters 1, 2, 3, and 4 appear in sequence, consistent with a traveling wave. However, progressive contraction becomes much more obvious when position along the ampulla at each time is transformed from 4 numbers to a curve by interpolation, and this curve is plotted against time as a surface in 3 dimensions. To aid in visualization, the surface is color-coded to indicate area, percent change from the mean, red positive and blue negative. There is a huge amount of information in this 350 seconds of data, but it is clear that the area oscillates with a fairly constant period, distance in seconds between peaks, but there are variations in magnitude along the length of the ampulla, 1 to 4. Note that grid lines of constant time cross the wave's surface at a considerable angle. To emphasize the progressive nature of the contraction, it helps to suppress some of the data by rotating the data surface until it is flat. It is now essentially a color-coded contour map. The middle contraction is the most regular, and I have drawn a straight line along the crest to emphasize the smooth progression of the contraction along the ampulla. Here I have made a color contour map showing contractions of ampulla 2. A 2D contour map is essentially the same as the previous 3D map seen flat on. I have made the grid lines dark so you can easily see that the peaks, dark green or yellow, tilt going from segment 1 at the bottom to segment 4 at the top. Thus the contractions move progressively along the ampulla as they did in the previous example. This pattern is most dominant in the middle of the map, 300 to 700 seconds. Progressive contractions could function as a peristaltic heart in a primitive animal. Of course, a real peristaltic heart is composed of a tube of muscle and contracts once a second, as opposed to once a minute as this one would. However, it's interesting to see that a nerve-based pacemaker isn't required to control a heart. Finally, I get to the main subject of this chapter. External force is detected when it stretches or compresses tissue. In this experiment, an instrument pushes on an ampulla to compress it. Pushing an ampulla in a controlled and reproducible way is not trivial. Remember, an ampulla is only 100 to 300 microns, 0.1 to 0.3 millimeters, wide and typically 3 times as long. My key instrument is a digitally controlled 4-axis micromanipulator. Here you see the control unit. Each of the knobs controls movement along one axis direction. X and Y define the plane parallel to the microscope stage, and Z is the vertical direction. D is the axis of the instrument that is being moved, which is adjustable. The position in these four coordinates is indicated in microns in the green LED window. Thus you can return to a predefined position with micron accuracy. The stepping motors that actually move the pusher are connected to the control unit by long flexible cables. Thus the knobs are mechanically isolated from the pusher itself. Force is applied to the ampulla by a thin glass capillary tube at the extreme left of this image. The glass tube is held by a 1/4 by 1/64 inch flexible brass strip attached to the 1/4 inch square brass beam in the middle of the image. The brass beam is held by clamps on the dark red rectangle, the actual positioning unit. The thin brass strip holding the glass tube is pushed down by a cylindrical magnet in the middle of a clear plastic bobbin from a sewing machine, holding a coil of 1,000 turns of number 36 wire with a total resistance of 64 ohms. 3 to 6 volts, drawing 50 to 100 milliamps, produces the force to depress ampulla. This current and voltage is easily controlled by an inexpensive transistor. On a rigid surface, the probe exerts a force of 0.33 nanonewtons per volt, but against a flexible target, the force would be less. Ampulla 2, a pair with 3, will be compressed for 20 seconds by the slender curved glass probe tip, which was pulled from the glass tube held by the pusher. The tip will be moved over the ampulla just before the push. The results will convince you that compression can alter the pattern of contractions, but will also raise many questions. There were 2 pushes in the 20 minutes of the full experiment. Here we see 5 minutes that brackets the first push, but it's speeded up 8x. Here is a frame from the scan of the video just before the probe was pushed down on the ampulla by applying 5 volts. The solid line of pixels is the edge of the ampulla defined by the scan. The probe is positioned about 5 microns above the tunic surface, above the ampulla. This is a frame after the probe moved down, in the middle of 20 seconds of compression. The edges of the ampulla have expanded 10 to 20%. This line graph presents areas of the 2 ampulla over 1,400 seconds. Ampulla 2 has been compressed for 20 seconds, starting at 486 seconds and at 926 seconds. In addition to these obvious features, the tops of peaks are broad and mostly flat, with a hint of small, fast oscillations. Finally, the peaks after a push are delayed by about 20 seconds, the length of the push, relative to the normal period of the contractions. Of course, you can't see this on this graph. A deeper insight into the response is obtained by dividing the ampulla into segments along its axis to define a line that changes shape with time, as we did previously in the analysis of a single detached ampulla. This is a 3D plot of that line in the 900 seconds that includes the two compressions. Time runs from left to right, and distance along the ampulla goes from segment 1 to 8, upper left to lower right on this 3D plot, and along the actual ampulla as seen in the photos. Starting at 300 seconds, left edge of the surface, there are clear contractions with a period similar to that seen after the pushes. To be sure, there are also many small, fast contractions, particularly in sections 1, 2, and 3, that complicate the pattern, and led me to call the contractions ragged previously. The peak at about 500 seconds, when the probe was actually compressing the ampulla, is also complicated, especially along segments 5 to 8. The peak after this push are complicated also, but in segments 1 to 4. It is these complications which make the top of the peaks appear flat. If you look at contractions in segment 6 during the 900 seconds, all peaks are quite simple, except during the two pushes. It's nice to see some simplicity in this complex plot. Segments 1 to 4 almost always behave in a complex way, and there is something strange going on in segments 7 and 8 after the two pushes. It's clear that the cells in this ampulla are only partially synchronized in their contractions, and some segments are much more rowdy than others. I could probably get a cleaner picture of the contraction pattern in a long, straight ampulla than for this rather short bean-shaped one. The position and shape of the probe may also be important. There's a whole summer of experiments to look forward to. I assume expansion, compression, and bending of the ampulla walls are the effectors of contraction, mediated by the molecular machines of the cell. Here, the original cross-section of an ampulla is the black circle, and the two progressive stages of compression are the green ellipse and the red bean-like shape. The main point of this diagram is to illustrate that compression by the probe can cause many progressive shape changes in the ampulla walls. Since our view is only from the top, we can't see specifically what is happening. We have seen the observed area of the pushed ampulla is increased during compression, which would be expected as it is flattened. However, the area of the partner ampulla actually becomes smaller, implying its volume becomes smaller, presumably because, counterintuitively, the volume of the pushed ampulla becomes larger by the push. If you look at the video of the push carefully, you can see cells flow into the pushed ampulla. Here are a few references if you want to explore what other people who study systems like these are thinking. The first two will give you an introduction to the known molecular machinery that is likely to implement the coupling between the detection of contraction and the generation of contraction, that is, the feedback loop that generates oscillations. I will warn you that a large amount is known about many molecular species, so these papers can be dense. As an example, there are so many species and accessories of the GTPases, and so many labs work on them that they have their own journal, and many GTPase papers are published in other journals as well. The last paper is an introduction to a two-dimensional Kuramoto model. Kuramoto models are very appealing, partially because applied mathematicians can solve many of them, or at least characterize their behavior. They describe a collection of oscillators that have the same amplitude and similar frequencies, coupled to each other only through their phases. These models have been shown to describe many world systems, and I think should be useful to model a single detached ampulla. I hope to demonstrate this in the next chapter in this series. In the next installment, Chapter 4, I will present models and simulations of contractions. Stay tuned.